Myself M. Gunakar, Assistant Professor of Zoology, Government Degree College, Siddipet, Telangana. Our today's topic is Hemichordata, General Characters and Classification. And at the end of this topic, we shall see the affinities of Hemichordata and its actual position in the animal kingdom. Now, let us look at the Hemichordata as an introduction. So, in the animal kingdom, some animal groups are referred as Invertebrates. This is a word belongs to some other language, which means of uncertain status. That means in the animal kingdom there are some animal groups are there whose status is questionable. That means under discussion. So the best example is Phylum Hemichordata. So today we are going to discuss this phylum Hemichordata. That means its status in the animal kingdom is somewhat doubtful. Now, why it has become doubtful? Let us see. Its other names are Stomochordata, Hemichordata. Some other names are also there, Stomochordata. The Stomochordata name is suggested by Davidoff and Adilochordata also. Then these hemichordates are deuterostomes. That means the blastopore becomes anus and they are enterocoelomates. Means mesodermal pouches that evaginate from the wall of the archenteron into the blastocele are fused with one another to form the enterocoelom. So here all the deuterostomes are enterocoelomates only, and so in the same way Hemichordata is a deuterostome and as well as enterocoelomate animal like that of the chordates and echinodermates. Formerly it was placed in chordates, but it was shifted. Now it is shifted to invertebrates. So for a long time the Hemichordata phylum was included under the phylum Chordata. Bateson in 1885 he has included this group in the phylum Chordata, and from that time onwards this group was under the Chordata, but in 1950 Silin proved that the notochord of these animals is neither homologous. That means it is structurally not similar to that of the chordates, nor analogous. That means functionally also whatever the notochordal structure that was proposed to be present or thought to be present in the hemichordates was neither homologous nor analogous to the notochord of chordates. Homologous means structurally similar, so it is not structurally similar, and analogous means even functionally is also not similar as per the Silin who has shown in 1950. Hence modern zoologists placed it under Invertebrata as the last phylum of Invertebrata, close to the Echinodermata phylum. Now being included under Invertebrata, the term Hemichordata becomes a misnomer. The term Hemichordata should be replaced by another suitable name in the future. So before going into the general characters of Hemichordata, here let us have a look at the other historical aspects here. Hemichordata has just now we have seen been removed from the Chordata and placed as an independent phylum of invertebrates close to Echinodermata. Bateson included the Hemichordata in the phylum Chordata, and but many French and German authors have separated it from the chordates, and Hyman has also placed it with the invertebrates. In the present system of classification, Hemichordata occupies a controversial position and exhibit no more than a convergent resemblance to the chordate organization. So now, with this background, historical aspects of this Hemichordata, let us have a look at the general characters of Hemichordata. So, Hemichordata, like that of the most invertebrates, they are multicellular animals. They are the deuterostomous. That means blastopore becomes anus. Just now we have discussed deuterostom and enterocoelomate animals. This is a small group of worm-like, bilaterally symmetrical and triploblastic and enterocoelomate animals. That means these are the worm-like and bilaterally symmetrical. The symmetry is bilateral symmetry, and triploblastic, that is presence of 3 germ layers, and enterocoelomate animals. We have discussed already what is enterocoelomate. Then these are exclusively marine animals. Not a single species can be found from freshwater habitats. Then these are commonly called tongue worms or acorn worms. Among these hemichordates, some of them are solitary. That means they are present in single, and most of them are slow moving animals. Whereas very few others are sedentary also, and they are colonial. That means sedentary means they cannot move, and colonial means they are present in groups. Very few of them. Then if we see the body is very soft and unsegmented. You can see here the figure, unsegmented. So the segments, as it is generally thought of whenever we see this figure of the hemichordates, we go to, it maybe strikes our mind that it is similar to the annelids, but annelids, as they possess segmentation, here no segmentation can be seen here in these animals. So body is soft, unsegmented, and divisible into proboscis, collar and trunk. Now you can see here proboscis, collar and trunk. So the first one, this is the proboscis, and the second one is the collar, and remaining portion up to here, all this portion is called as trunk. So the body is very soft and unsegmented and divisible into proboscis, collar and trunk. Then body wall, if you see, the body wall is covered by a single layered epidermis, and below that a basement membrane is present. That means on a basement membrane a single layered epidermis is situated, and then below the basement membrane we come across longitudinal muscle layer is also present. Then if we look at the coelom is, as we have already discussed, is enterocoelous coelom, which is divided into 3 regions. That means the coelom, and the coelom that is present in the proboscis is called proboscis coelom, collar coelom and trunk coelom respectively. That is, the coelom is not continuous throughout the body, it is divided into 3 divisions of the body, corresponding divisions of the body. So coming to the next general characters of hemichordates, the digestive system, physiological systems, digestive system. So here in the digestive system of hemichordates, a complete alimentary canal is present from mouth to the anus, and this alimentary canal again divided into foregut, midgut and hindgut, and it is, in some animals it is straight, or in other animals it may be U-shaped, and it opens outside through anus. Then most of the hemichordates, they feed on microorganisms and organic debris. The proboscis consists of an outgrowth from gut, which is called a buccal diverticulum or stomochord. So because of this structure which is present in the proboscis, which was regarded as notochord in the past, that's why Bateson in 1885 and other scientists has placed this Hemichordata in the Chordata phylum as a subphylum. So, but recently it was found that notochord, the buccal diverticulum or stomochord, which is present in Hemichordata, is as per the Phylum in Phylum, it is neither homologous to the notochord of Chordata or analogous to the notochord of Chordata. So that's why the most of the scientists tilted towards the shifting of this Hemichordata subphylum to the Invertebrata. Then coming to the respiratory system. Now the respiratory system here, one to many pairs of gills are present. In some animals only one gill is present, but most of the animals possess many pairs of gills are present, and these gills are opened outside by gill slits. Now coming to the circulatory system, the circulatory system possesses a dorsal heart. Heart is situated on the dorsal side, and two longitudinal blood vessels are also present. So please remember these characteristic features. Dorsal heart is dorsal in all invertebrates, whereas heart is ventral in the chordates. So based on this characteristic also, Hemichordata has more affiliation to the invertebrates. Then two longitudinal blood vessels are present, and these longitudinal blood vessels are a dorsal longitudinal blood vessel and a ventral longitudinal blood vessel. These two blood vessels are connected by many lateral vessels. Coming to the excretory system has a single proboscis gland in some animals, whereas in other animals the glomerulus, which is supplied with blood vessels, and the blood comes to the glomerulus where the nitrogenous wastes are filtered and excreted outside. Then nervous system is very less developed. It is diffuse and consists of nerve cells, nerve fibers, and dorsal and ventral longitudinal strands are present, which are connected by circular strand in collar area. Then reproductive system. As far as reproductive system is concerned, the animals are unisexual. That means sexual dimorphism can be seen. Males and females can be seen in some animals, whereas in some other animals are bisexual. That means hermaphrodite. That means male and female sexual reproductive organs are present in a single animal. There is no sexual dimorphism in some other animals. Whereas gonads are one to many parts. That means in some animals gonads are only one, whereas in some other animals gonads are many pairs are present. Then fertilization, if you see, the external fertilization is present in all animals, and development is indirect development, that is free swimming larval stage is present, and this larval stage is named as Tornaria larva. In some development is direct also. So in some animals where development is indirect, Tornaria larva can be seen. In other animals development is direct, where no larval stage is present. Now coming to the Hemichordata classification. Now Hemichordata, as though it is a very small group, as it is possessing many different characteristic features, it is classified into 4 classes. So Phylum Hemichordata is classified into 4 classes. Class number 1 is Enteroneusta, number 2 Pterobranchia, number 3 Graptolitha, and number 4 Panctospiroidea. I repeat: class Enteroneusta, class Pterobranchia, class Graptolitha, and class Panctospiroidea. And under Enteroneusta, Balanoglossus and Sacoglossus are placed. Whereas here under the Pterobranchia, this Pterobranchia is again divided into 2 orders. Order number 1 is Rhabdopleurida, and order 2 is Cephalodicida. And under Rhabdopleurida, the best example is Rhabdopleura, and Cephalodicida has the Cephalodiscus as its example. Now under the Graptolitha, the Dendrographus is the best example. So Panctospiroidea, the example is Panctospira. So this is the classification of Hemichordata. Now let us look at the characteristic features of each class. The Hemichordata, first class, as we have discussed just now, is Enteroneusta. So these Enteroneusta all are free living. That means none of them is a parasite or commensal. So all are free living animals. They are solitary. Solitary means they live in singles, and they are burrowing forms. That means they make burrows on the in the debris, that means on the floor of the oceans and seas, and they live in those burrows, that is they are fossorial. The body is long and worm like. Now you can see here the body also, the shape of the animal also. Then proboscis tapers anteriorly. This is the proboscis which you can see in the green color, made in the green color. Actually the animals, the color is, I have made the color, but actual color is maybe different. So this proboscis is tapering towards the end. It is sharp at the end, and many pairs of gill slits are present. Just now we have seen in this some of the animals, hemichordates, in some of the animals gill slits are less in number, and many pairs are in some other. So many pairs of gill slits you can see here in this figure also. We can see on the body. So here the gills, now gill pores you can see here, external gill pores. So many gills are you can present here. So that's why many pairs of gills are present in these animals. They open outside by gill slits. Then these animals are unisexual, that means male and female are distinguished. Their sexual dimorphism can be seen, and gonads are numerous, whereas in some other animals of the Hemichordata gonads are single, but here numerous gonads are present. And when we see the development is indirect, possessing a Tarnaria larva in the life cycle, and best example are balanoglossus and saccoglossus. Now you can see the figure of saccoglossus here. This is the saccoglossus figure. Almost it is similar almost to the balanoglossus with slight variations. So here even in the balanoglossus also the same structure can be seen. That is, the body is divided into proboscis, collar and trunk. Here also proboscis, collar, and the remaining portion up to here is collar. So in almost similarity can be seen in the balanoglossus and saccoglossus. Both are included under first class Enteroneusta. Now coming to the second class, Pterobranchia. Pterobranchia has many groups, many animals here as examples. The best example is Rhabdopleura. Then what is the structure, or what are the features? Let us see. They are sedentary animals. Just now we have seen the Enteroneusta. They can move, that is they are burrowing, that means they are worm-like and they can move. But here these are sedentary animals. Pterobranchians are sedentary, they cannot move, immovable animals, and these are colonial animals, whereas Enteroneusta are solitary animals. These are tube-dwelling animals, whereas Enteroneusta are fossorial animals. These are tube-dwelling or tubicolous animals. Body is short and flower vase-like shape. Here you can see the body is, in the figure you can see. Then proboscis is shield shaped. Gill slits, only one pair of gill slits are present here, when compared to the Enteroneusta, where many pairs of gill slits are present. Then the animals, these when we see the reproductive system features, they are unisexual or bisexual. Here also Heterobranchia, some of the animals belong to Pterobranchia are unisexual and some of them are bisexual. That means sexual dimorphism can be seen in some, and no sexual dimorphism, but both the sexes are present in same individual, as in like that is bisexual. Then development is direct or indirect. In some animals the development is direct without any larval stage, and in some indirect development also can be seen with a larval stage. And here one more feature which is very much important is, here these animals, the Pterobranchians, they undergo asexual reproduction also. They undergo asexual reproduction by budding. So generally this asexual reproduction generally animals undergo whenever the sexual reproduction is not possible. So they undergo asexual reproduction, they produce offspring by budding also. Best example is Rhabdopleura. Now coming to the next class, Phylum Hemichordata. There are 4 classes just now we have discussed. The third class is Graptolitha. Now here you can see the Graptolitha. So what is the specialty of this Graptolitha is, they include only extinct and colonial animals. They are the extinct animals. So how we know that these Graptolitha animals, then if they are extinct, that means we have a fossil records. We have a fossil records, and that's why we came to know that these animals are belonging to Hemichordata, and they have to put under a separate class. That's why a class is made here under the name of Graptolitha, and they were found in Ordovician and Silurian periods. Ordovician and Silurian periods of Paleozoic era. Then best example of this is Dendrograptus. Now coming to the next and last class of the phylum Hemichordata is class Planktospiroidea. And these animals are known only by pelagic larva related to Tarnaria. Only by pelagic larva that is related to Tarnaria, and their adult forms are not yet revealed, and nobody knows the adult forms. So adult forms have to be discovered. They are yet to known. Then what is the specialty of this digestive system is alimentary canal of larva is U-shaped. But this U-shaped larva becomes straight or it remains U-shaped in the adult animals. We don't know, because the adult animals are not yet known to anybody. Then coming to the ciliated bands are present in larva. Some of the branching ciliated bands are present in the larva. The best example is Planktospira. Now coming to the, so we have seen so far the Hemichordata characteristic features and the classification, and in the classification various classes and orders and their examples we have seen. So let us go to the affinities of Hemichordata with the various animal groups. So in the first instance we have discussed that the Phylum Hemichordata or subphylum Hemichordata as a group has many doubts regarding its present status or position in the animal kingdom. So scientists have searched for the affinities of Hemichordates with the various groups of animals. So lastly they found that the Hemichordates have some affinities with some invertebrate groups, in addition to the chordate groups also. So main invertebrate groups, the Hemichordata has affinities are with Annelida as well as Echinodermata. Let us start with the affinities of Hemichordata with the Annelida. Now first of all let us see the similarities of Hemichordates and Annelids. So first and foremost similarity is body is worm-like. Just now we have seen in the case of Saccoglossus and Balanoglossus in the phylum Hemichordata, which is a body is worm-like. So annelids are also, they are the worms. Then burrowing. The burrowing feature we have seen in the case of Entitonusta, and not only burrowing, they are ingesting mud like that of the most of the annelids like earthworm. Then proboscis is homologous to the prostomium. We have seen in the annelids, prostomium is the striking structure that is present in the before the first segment or before the mouth. So here the proboscis of this Hemichordata is homologous, that is structurally similar to with the prostomium. Now coming to the next characteristic feature, the heart is dorsal. Like that of the most of the invertebrates. Here annelids where heart is also dorsal, and here the dorsal heart and longitudinal blood vessels are also present. Even in the annelids also longitudinal blood vessels, dorsal blood vessel, ventral blood vessel and other blood vessels are also present. Here also dorsal heart and longitudinal blood vessels are present. Then Tarnaria larva, which is present in the Hemichordata, is similar to Trochophore larva of annelids. So all these similarities suggest that there is affinities with the Hemichordata with the Annelida. But there are dissimilarities also. They are not less than the similarities. So let us have a look at the dissimilarities also. So presence of gill slits and buccal diverticulum. So where here Hemichordates possess gill slits, and even annelids the gill slits are not at all present, and buccal diverticulum which is present in the proboscis of Hemichordates is not present in the annelida. Tubular dorsal nerve cord. See here, tubular dorsal nerve cord. See, tubular, that means the cavity is present, dorsal nerve cord is present, it is a cavity like structure, but here in the annelida, though nerve cord is ventral and as well as it is solid. In the annelids, nerve cord is double ventral nerve cord is present and it is solid, whereas here nerve cord is dorsal and it is also tubular. Then nephridia are present in the annelids, whereas no nephridia are present in the Hemichordata. Just now we have seen in the structures, when we see the different characteristic features of the Hemichordata, we have seen that there is a under the excretory system, the single proboscis gland or glomerulus is present, which is connected with the blood vessels, but here no nephridia were present in Hemichordata, but nephridia are mainly the excretory organs in the annelida. This is also a main striking difference. Then blastopore becomes the anus in Tarnaria. So whereas that means here the deuterostomous condition is prevailing in the Hemichordata, whereas annelids are protostomes. So here the blastopore becomes the anus in Tarnaria, that is belongs to Hemichordata, whereas in Trochophore the blastopore becomes mouth, as annelids are belong to protostomes. So these are some of the dissimilarities, and these dissimilarities are more when compared to the similarities. That's why, though the Hemichordata is possessing some similarities, we can't place or we can't connect the Hemichordata with annelids. Now coming to the affinities of Hemichordates with Echinodermata, other group Echinodermata. Now when compared to the annelids, Hemichordates have no similarity of adults in both the groups here. We need to know that no similarity of adults of Hemichordates and Echinoderms. Then if at all there is a similarity is there, that is only in the larval stages, and that too in the organization of nervous system, which is poorly developed in both the groups. And one more thing is same habitat, that means echinoderms are all exclusively marine and Hemichordata are also exclusively marine, and power of regeneration is also most striking in both the groups. Both are enterocoelomates here, only. That means coelom, how the coelom is formed in enterocoelom, mesodermal pouches evaginate from the wall of the archenteron into the blastocele, which are fused with one another to form the enterocoelom. And here larval stages of both the groups show many similarities. So here just now we have discussed no similarity of adults, but larval stages show both the groups show many similarities. Then that's why Fell, a scientist in 1963, believed that both the groups might have evolved from a common ancestor. Now coming to the affinities of Hemichordata with the Chordata. So far we have discussed affinities of Hemichordates with the annelids and affinities of Hemichordates with the Echinoderms. So both of them are having differences as well as similarities with both the groups. Now coming to the affinities of Hemichordata with Chordata, now let us see the similarities. Three fundamental chordate characters, that means presence of notochord, presence of tubular nerve cord, and presence of gill slits, presence of pharyngeal gill slits. These are also present in Hemichordates. So this point gives very boost to the scientists who discuss that Hemichordates are a group belong to the Chordata. Now second one is Buccal diverticulum is regarded as notochord. Just now we have discussed Buccal diverticulum, which is present in the proboscis region of the Hemichordates, was regarded as notochord. Then dorsal position of collar cord similar to dorsal tubular nerve cord of chordates. So in the collar there is a cord-like structure is present, that is called collar cord, which is dorsal in position, and it is this dorsal nerve this is similar to the dorsal nerve cord of the chordates. And we have seen that many pharyngeal gill slits are also present in the hemichordate. These three main characters, they show that the hemichordata may be placed under chordate. Now coming to the dissimilarities. Just now we have discussed that the hemichordates are having all three primary or diagnostic chordate characters. But there is a striking difference, especially with the notochord. What we have discussed so far is the buccal diverticulum that is present in the proboscis is homologous to the notochord. But here you can see, if we see these structures, if we see the differences here or features here, you come to know that why buccal diverticulum cannot be called as a notochord. Now first one is buccal diverticulum is formed as a forward evagination of the buccal wall, whereas notochord is formed as a solid rod-like elevation from the roof of the archenteron. So this is a striking difference. Then second one is the buccal diverticulum is made of ordinary epithelial cells, whereas notochord consists of large vacuolated cells. Then buccal diverticulum is also not having any sheath surrounding it, whereas notochord is covered by a sheath. Then the buccal diverticulum is not at all related to blastopore, but notochord is related to blastopore. Then the buccal diverticulum do not support in function of the animal, whereas notochord is supporting in function. It provides strength to the animal body. Then buccal diverticulum is ventral to the mid-dorsal blood channel, whereas the notochord is always dorsal to the main dorsal blood vessel. Now these are the differences. And some sort of prioral gut confined to the proboscis. That means it is buccal diverticulum is a prioral gut, some sort of prioral gut confined to the proboscis, but not the actual notochord. So here notochord is axial stiffening rod extends far backwards, whereas buccal diverticulum is subjected to only proboscis, whereas here notochord is strengthened extends up to far backward of the body of the chordates. Now with these differences, especially with the notochord and buccal diverticulum, now we can come to a conclusion that notochord is absent in hemichordates. Whatever that was thought of notochord, that is buccal diverticulum as a notochord, is not at all correct. The nervous system is typical invertebrate pattern. Just now we have seen nervous system, how it is. Nervous system, invertebrate pattern. No post-anal tail is present, and blood vascular system is invertebrate type. Blood vascular system, because heart is dorsal, and the dorsal blood vessels are also longitudinal blood vessels and connected by the lateral blood vessels. All these shows the blood vascular system is invertebrate type. Now with this discussion of the affinities of hemichordates with all these three groups, that is annelids, echinoderms and chordates, now we come to a conclusion. So what is the conclusion? Let us see. The resemblance of hemichordates with both the chordates and echinoderms shows that all the three groups, that is hemichordates, echinoderms and chordates, are related to each other. From embryological, biochemical and other serological studies, it was proved that it may concluded that the three groups have a common evolutionary line, which were possibly evolved from Dipleurula or Tarneria like larva. And the hemichordates evolved from this ancestral line, after the divergence of ancient echinoderms. That means first of all the ancient echinoderms might have evolved, and after that these hemichordates might have evolved, and but these hemichordates definitely would have evolved before the rise of the true chordates. So they are often described as pre-chordates. That means before the evolution of chordates, these hemichordates might have evolved, but after the evolution of ancient echinoderms. So that's why these hemichordata is the group as a small group can be placed under the invertebrata between between that means after the echinodermata, but close to the chordates as well. So it can be named as pre-chordates, but it cannot be placed under chordata because of the striking differences that has shown by the hemichordates so far we have discussed. So this conclusion can be drawn from such discussion. Thank you.